Welcome everybody. Welcome to Greg Dore, who's our visiting Scientia Professor today. Greg will be known to many of you. He's an expert in hepatitis C and also HIV, but particularly hepatitis C, virology and epidemiology. Greg works at the Kirby Institute. He leads a large research team there. He's been very influential in a lot of the efforts to eliminate hepatitis C around the world, and particularly, I think, leading a lot of the Australian effort for that. He's been very involved in a lot of the prison studies and leads a large group, and I think has been a really, as I said, a very prime driver for the implementation of all the direct-acting antivirals and the efforts to eliminate hepatitis C in Australia. So it's a great privilege and pleasure to have Greg talk to us today. So I'd like to introduce Professor Greg Dore.
Thanks very much, Mark, and thanks to Ben for the invitation to present. Just let me share my screen.
Thanks, Greg. We can see. That's excellent. Yeah.
If I'd firstly also like to acknowledge the traditional owners of the land that I'm on, the Bidjigal people here at Kensington at the University of New South Wales, and pay my respects to elders past and present. Today I'm going to give you an overview of the life that I've led professionally over more than a couple of decades, involved in hepatitis C therapeutic development, particularly with a focus on people who inject drugs. These are my disclosures. So I'll try and cover different aspects of my personal involvement in hepatitis C care for people who inject drugs, the interface between clinical care and therapeutic research, which I think is a really pivotal aspect and certainly has been central to my professional work. Give you a little bit of insight into some of the investigator-initiated hepatitis C therapeutic research protocols that we've been involved in developing and actioning, and specifically look further into Hep C treatment as prevention and the context it provides for Hep C elimination. And at the end, a little bit of discussion that may come up in the Q&A as well, in terms of the role of community engagement and advocacy. As mentioned, I'm at the Kirby Institute. I actually filled out a survey this morning. The Kirby was surveying different aspects of their staff, and you had to give how many years or months, if it was less than a year, you'd been at the Kirby Institute. And I had to give a number of 28 years. I don't know whether that's a sign of loyalty or inertia. But it certainly has been a privilege to work at the Kirby. I've been leading the Viral Hepatitis Clinical Research Program for many years now, and it's one of many programs we have at the Kirby across HIV, viral hepatitis, sexually transmitted infections, and different aspects of those disease areas. I just want to take you on a bit of a journey in terms of therapeutic development. If you think about Hep C treatments, you can think about them in the context of how well they're tolerated and how effective they are. Way back in 1997, I gave a presentation at the first Australasian Hepatitis C conference. It was held in Sydney. It was about Hep C treatment, and I somewhat provocatively stated that no one should be treated with hepatitis C. At that stage, we only had interferon monotherapy approved on PBS. The cure rate was around about 10%, and even those people that were cured, their predictors were also predictors of non-progression of liver disease. So I really didn't feel that we were doing much in terms of trying to reduce the burden of hepatitis C, and we had quite a toxic therapy. We had the addition of ribavirin and oral therapy to interferon injections became available late 1999 through compassionate access, and that's when I set up the first hepatitis C clinic at St Vincent's Hospital. At that stage in 1999, there was a requirement for a 12-month period of abstinence from injecting drug use. My one, I suppose, time of professional civil disobedience was to ignore that requirement. And in fact, the second patient I treated was still injecting. And I think that sort of one decision back in late 1999, early 2000, to look at people who inject drugs as individuals requiring high quality care was something that really set me on a pathway in terms of broadening access to this incredibly marginalised population that we have, who had clearly a high burden of hepatitis C. Since that time, in terms of the research world, we've had three NIH R01 grants, 5-year grants, to look at acute hepatitis C therapeutic development. That's been one of our main areas of research. We collaborated with the Hepatology Network. This is one of the only studies, in fact, that I've been involved in through the Kirby Institute, which hasn't been an investigator-initiated study. So this was a study that was developed by the Hepatology Network and came to us to undertake the operational side of the Chariot study. All the other studies that I've mentioned here as we moved into the direct-acting antiviral therapy era were initiated by investigators within our program at the Kirby, in terms of the ideas, obviously working with a collaborative network of sites, working with research partners, and leveraging both private and public sector funding. So it's been an amazing sort of journey over the last couple of decades, a real privilege to be involved in Hep C therapeutic development. I mentioned at the outset some aspects of the interface between clinical care and therapeutic research. Also, I think it was important in terms of the breadth of my involvement. I said I set up the first hepatitis C service at St Vincent's Hospital in 1999. I then opened services within the methadone clinic, so the Rankin Court Drug and Alcohol Service in the early 2000s. So I was involved in setting up Hep C care there. And then I was also involved in setting up the first hepatitis C clinic at the Kirkton Road Centre shortly thereafter. So I've had involvement at the tertiary level, including within the drug and alcohol service, and also at the community level. And I think this sort of diverse clinical involvement has really given me an important foundation for being involved more broadly, both in care and in research. It's interesting to look back over some of the key sort of studies that we're involved in. This was the first study where we wrote up that experience of treating people who had ongoing injecting drug use. They were relatively small numbers, but there weren't many studies at the time. We collaborated with the group at Western Hospital in Melbourne and wrote up our experience. And one of the first studies to describe outcomes of people who continued to inject. We then moved into acute hepatitis C. I mentioned we had an NIH R01 grant, and this was the first sort of study, interferon-based therapy of treatment of acute hepatitis C, with really quite favourable outcomes, despite a very marginalised group with frequent injecting drug use. And then we moved into the direct-acting antiviral therapy era after we established an international network of sites that could enrol patients with ongoing injecting drug use. So if you think about this development, it was really going from a local experience at the sort of level at Darlinghurst at St Vincent's Hospital, moving into a national network of sites that could recruit patients with acute hepatitis C to undertake pivotal studies that obviously US was very interested in, because they funded us through the NIH to do these studies. They felt that they couldn't be done similarly in the United States. And then to an international sort of phase of developing site network across many countries around the world. I also had involvement with industry. Some people get concerned about involvement with industry. I thought it was really important to work with the key industry groups who were developing Hep C therapeutic agents. They provided access to patients at the tertiary level for some of these newer agents. They also allowed specific clinicians to take leadership. And we worked very closely, for example, with Merck to develop the first randomized controlled study of direct-acting antiviral therapy in a population who were on opioid agonist therapy, many of whom were still injecting. The first sort of study to include people with ongoing injecting drug use. I've also been involved in broader population studies with some of these new agents. I mentioned we set up national networks initially for ATTACK, the acute Hep C trial, but a range of different initiatives they're undertaking in terms of research in the community, in the prison setting, in people with hepatitis C monoinfection, those with HIV co-infection. We also, as I mentioned, then moved into a phase of international development with sites largely in Europe and North America, but some other sites in Asia in terms of studies of both acute and chronic hepatitis C, including some studies specifically among people who inject drugs, which was our sort of core business, for example, but also a study such as SMART-C, which was looking at whether you needed monitoring of virological response and adverse events during treatment through clinic visits or not. And that was a population of people that weren't injecting, so it's a broader general population. So we did do studies outside of the injecting drug user population. I wanted to show some findings from a couple of our pivotal studies, Simplify and Defeat. And they were relatively small studies, but many of the key studies for direct-acting antiviral therapy were relatively small in terms of population size and often single arm as well. So not all the sort of pivotal or even the licensing studies were randomized controlled studies, given how effective these therapies appeared to be. So Simplify and Defeat recruited through a similar international network. And I'll just show you a couple of sort of outcomes. The study design was very similar, 12-week regimen. The two regimens, sofosbuvir and velpatasvir, then the AbbVie PROD plus or minus ribavirin sort of regimen delivered over a 12-week period with the usual study follow-up. But the key difference to the study follow-up, we followed people for their treatment outcome, but then we had continuing follow-up 6 monthly for incidence of reinfection, clearly an important secondary outcome in a study among people who inject drugs. Just wanted to compare these sort of three key studies. So the study I mentioned before, the Merck study, was called COSTAR. And it's interesting how things evolve. So the industry was a little bit nervous about including people who injected drugs. So they wanted a pretty stable population for the COSTAR study. So the first study of its kind. So everyone in that study had to be on opioid agonist therapy, was considered therefore to have greater sort of stability, less issues in terms of adherence to the regimen. But importantly, they did include people with recent injecting drug use. Then we moved to our two investigator-initiated studies, so the Defeat study, where we included populations of people that were on stable opioid agonist therapy, whether or not they were injecting. But we included also people that were injecting, but not on OAT. And then the Simplify study was sort of pushing the envelope even further. So we wanted to do a study within a population where everyone had recent injecting drug use. So we included some people who are on stable OAT, but we didn't include individuals who are on stable OAT, but not injecting. And you look at the proportion of people with recent injecting over the previous 6 months, so 25% for the COSTAR study, up to 57% for the Defeat study, and this is 100% of people with recent injecting in the Simplify study. So I think an important sort of evolution of studies looking at somewhat different populations within the broader population of people who inject drugs. Just combining the Simplify and Defeat studies, this was a marginalised population with a significant proportion with unstable housing, high levels of hazardous alcohol use, and high levels of injecting even within the last month. So the criteria was within the last 6 months for Simplify and or on OAT for Defeat. You can see here the proportions of people on OAT with or without recent injecting. So we felt a pretty sort of representative population of people that injected drugs. And then you go to the cure rates. I mean, these therapies were amazing, and all of us who've been involved in Hep C care over the last sort of decade or more has been privileged to see the paradigm shift that we've had going from interferon-based therapies to the all-oral direct-acting antiviral therapy regimen. So the COSTAR study, 92% of people cured, Defeat 91%, and Simplify 94%. And these are intention to treat cure rates. So given these populations, particularly with Defeat and then with Simplify, people with high levels of ongoing injecting drug use, we felt really impressive cure rates given that loss to follow-up equals non-response in an intention to treat analysis. We also took the opportunity, because we felt adherence was going to be an issue, to monitor it very, very closely. So we had electronic means to monitor adherence. So all the regimens were in electronic blister packs. So whenever a patient took the medication out of the blister pack, it sent a signal, and that was captured. So we had a record of that being undertaken. Obviously, it doesn't guarantee the person putting that pill in their mouth and swallowing it, but we're pretty sure that the vast majority of cases that happened. So you can see here, each individual in this figure is a row, and the light blue patches where patients didn't take the medication on specific days. So we had a really detailed record of all individual patients and the days that they took medication and the days that they didn't. With the once daily regimen of sofosbuvir and valpattizivir, 94% overall adherence, a little bit lower, interestingly, in the twice daily regimen. But if you think back to the cure rates of above 90%, despite some levels of non-adherence, you can see here individual patients not having therapy for a week or more, and then recommencing therapy, despite that non-adherence level, really favourable outcome. So demonstrating a sort of level of forgiveness with these regimens. We also looked at injecting drug use behaviour throughout the treatment and follow-up, and it was relatively stable. So we did see some reduction in the proportion of people injecting opioids over time in the combined sort of studies. But we expected there to be ongoing injecting drug use despite engagement with clinical services, despite counselling about harm reduction, etc. So we weren't concerned about that. We just wanted to monitor what the level of injecting drug use. We were obviously trying to reduce the proportion of injecting that was with shared injecting equipment. You can see here a somewhat reduction in the proportion of people that reported sharing needles and syringes over the past month. So that was encouraging to see. And hopefully that would translate into a lower reinfection incidence. Moving on to another study that has been really a landmark study, I think, for our research program at the Kirby and internationally really looked upon as a key study, was the STOP study. So this was the study to evaluate hepatitis C treatment as prevention with the new DA regimens. We partnered with Gilead in terms of drug provision, but this was investigator-initiated. We had some funding through an NHMRC partnership grant. It was co-led by myself and Andrew Lloyd. And we chose 4 prisons in New South Wales, Goulburn, Lithgow, and 2 prisons in Sydney. So 2 maximum security prisons, Goulburn, Lithgow, and 2 medium security prisons, including 1 female prison. We wanted to monitor the incidence of hepatitis C and then scale up treatment rapidly and look at the impact that treatment scale up had on incidence. So we enrolled 3,600 inmates across those 4 prisons. So a huge undertaking in terms of enrolling and following a very, very large number of participants in this sort of a study. In fact, we reached coverage levels of around about 60% across the prison. So we had great engagement within the prison population. We, in fact, employed some prison officers to assist with that engagement. There was initially some reluctance from the prison officers to be involved in this type of research, and really they weren't terribly overjoyed by a lot of money going towards prisoner health. But I think we really sort of brought them around to how important this initiative was. We also mentioned that maybe they would have a reduced occupational health risk related to hepatitis C if we're able to reduce the amount of virus circulating in the prison system. And everyone that we enrolled was irrespective of whether he had a history of injecting drug use. We thought it was important to not just focus on people who reported injecting drug use because of the potential issues around reporting. We screened everyone for hepatitis C. Those that were antibody positive, we then tested for Hep C RNA. So we came up at baseline with 3 groups. So those that were clearly infected, antibody and RNA positive, those that had evidence of previous infection, but not active infection, that can be either through spontaneous clearance or treatment-induced clearance, but these people at risk of reinfection, and those that were uninfected being antibody positive. But we wanted to follow all 3 groups. Obviously, this group, we wanted to refer for treatment, and we wanted to follow these 2 groups for incidence of new infections. Started very late 2014, really started in 2015. And we had this phase of surveillance of monitoring the incidence of Hep C infection, and then a rapid treatment scale up from mid-2017. During this initial period, you could be treated through the normal sort of channels, which initially were interferon-based, and then with PBS inclusion of DA therapies from March 2016, inmates could receive DA therapy prior to the scale-up. If we look at the scale-up, though, most of the treatment happened, as I said, from sort of mid-2017. So you can see here that numbers of people treated through the normal prison health service, and then treatment through STOP-C with the scale-up. So a very rapid sort of increase in treatment during 2017. And that led to an encouraging reduction in incidence. This is total incidence. You can see the incidence prior to the scale-up, in fact, increasing, and then this decline following the rapid scale-up. And that was both in terms of new primary infections and reinfection, less impacted, although on the reinfection side. And when we looked at the Hep C incidence pre-scale-up, it was 8.3 per 100 person years, reducing to 4.5 per 100 person years. So about a halving of the overall incidence significantly. So a really important demonstration, we felt, of the impact of treatment as prevention. The new phase in terms of elimination is all about getting people diagnosed rapidly, getting them linked to care and onto treatment. And we've been leading, particularly Jason Grebely, within our program, initiatives around point of care testing. In fact, we did the initial field evaluation for the Cepheid expert Hep C viral load. We did the lobotomy-based evaluation, and then we did the finger stick-based evaluation. So we've been fortunate to be working very closely with industry in terms of evaluating these new diagnostic tools, and then to do important implementation, science-based sort of research. Wanted to just show you a couple of studies. We did a pilot study in the community where we set up at a community-led needle and syringe program, and we did initial screening with finger prick RNA. We also tested for HIV and hepatitis B through finger prick testing, a nurse assessment, peer-based support, and our goal was to try and get people onto treatment as quickly as possible. So a relatively small study, found 27 people who are Hep C RNA positive, 20 of those 27 initiated onto treatment, a couple outside of the tempo study. So overall treatment uptake of 81%. So very encouraging. And most people initiated treatment within a few days, many people on the same day. We did a further pilot study in the prison setting called Pivot at Kempsey, where Andrew Lloyd was leading this study. We had a control period where we monitored screening and treatment uptake for 3 months, and then introduced, again, a one-stop shop intervention with Hep C screening and initiation of therapy. So in terms of RNA detection and testing, increasing from 18% of RNA testing to almost 100%, as you might imagine, given the one-stop nature of it. And then treatment uptake, 26% in the control period to 93%. And 26% of a smaller sort of denominator. So obviously a large number of people weren't tested for Hep C, antibody and RNA, could have had an active infection. So I think pretty clear evidence that this intervention works and is really the solution to getting people onto treatment. We've moved into a new sort of phase with a national sort of program, again, led by Jason Grebely, the National Point of Care Testing Program. And this is on the back of us being involved in point of care testing for sexually transmitted infections and COVID-19 through a very large Indigenous community network across the country. So each green dot is a community where we have a point of care machine, where we've been, as I said, testing for sexually transmitted infections in COVID for some time. So it's been great to have that sort of foundation. So this is the National Hep C point of care testing program with the current sort of sites. We're expanding the network. We're looking to be in 90 different sites, planning to screen between 50 and 60,000 people, sites in both the community and the prison setting. There's a lot of work to be done in terms of the logistics of this sort of screening, the connectivity, how you report the positive results that haven't been previously notified through public health surveillance systems, how you sort of link in with laboratories in terms of quality assurance. So it's an enormously complex project, but I think a really key sort of project. We're also looking at not only Hep C RNA point of care testing modalities, but also new Hep C antibody testing. So, for example, we're evaluating a 1-minute Hep C antibody point of care test. The previous one took up to 15 to sort of 20 minutes. So we're looking at whether we can get that down. And a lot of settings, if they don't have expected high Hep C RNA prevalence, it's much more cost effective, and we've done health economic sort of work around this to do an initial finger prick antibody test, particularly if it only takes a minute or two, and then to sort of reflex to point of care RNA testing for those that are antibody positive. So we're evaluating different strategies around the sort of sequencing of Hep C point of care testing. And this is where we're up to in terms of the overall rollout of this initiative. So 59 sites enrolled in the program, 17 of those prison sites, over 8,000 point of care tests done, predominantly RNA, but increasing number of antibody tests being done as well. It's interesting if you look at the breakdown so far in the prison side of things, where 2/3 of the testing has been done, RNA prevalence in the tested population, 15%, in the community, 11%. We're then looking at what the treatment initiation is overall, pretty amazing. 86% of those who are RNA positive were treated within a 12-week sort of follow-up period. And that's driven by incredibly high treatment initiation in the prison, where it's 93%. We're still working through some issues on the community side. We only have 51% treatment initiation, so trying to sort of remove any further barriers to treatment initiation through the community sites. And it's early days, so I expect this treatment proportion to continue to improve over time. A great sort of initiative that the Kirby is involved in, and I think one of the sort of hallmarks of the work that we've done is working very closely with community. All of our sort of major sort of studies in recent years have had strong peer support within our teams. So we send out teams for the previous ethos study and now the National Point of Care Testing Program. We have a team of people from the Kirby. They have a peer worker to work with them, really improves engagement with the client sort of base. And it's sort of a fun activity for our researchers to be involved in as well. And as I said, huge undertaking, lots of different sort of partners at the sort of site, at the government, at the stakeholder sort of level as well. Just wanted to move on to some aspects of broader Hep C elimination in Australia. Many of you may have seen this figure before. This is the amazing transformation of Hep C treatment in Australia from an era when we were sort of struggling along, treating a few thousand people a year with interferon-based therapy. The patients were struggling a lot more than the clinicians with the 6 months up to sort of 12 months of interferon-based therapy and the side effect profile that came with that. We had some improvements, but really no major sort of changes to really start to make a dent in terms of Hep C elimination. It was really late 2014 we started to get a trickle of early access to these new therapies through compassionate access programs, some generic importation through 2015, some clinical trial access, and March the 1st, 2016 was our launch day for sort of PBS funded DA therapy. It's incredible, more than 30,000 patients treated in that first sort of 10-month period from March to December in that year. There's, as expected, been reductions since then, but we've treated around about 100,000 people with DA sort of therapy over that period. 7% of patients have been retreated. A really important aspect to the government-funded program when we were developing it was to make it as broad as possible in terms of people that could access the therapy, broad in terms of the prescribers that could be involved in treatment, and also to acknowledge that some people would fail therapy, not just for virological failure, a small proportion, but also people with ongoing risk behavior would be reinfected. And we wanted to ensure that they would have access to retreatment, and not just one course of retreatment. People needed 2 or 3 courses of retreatment. That was also available, and to really not have any judgment in terms of people becoming reinfected, to deal with it as an infectious disease as it should be. As I said, really broad and diverse models of care, which I think has been pivotal and underpinned by several different and consensual national hepatitis C strategies that bring together stakeholders in partnership to develop, really, I think, world-leading strategies. Just a bit of data showing the change in pattern of prescribers. This only goes back to 2019, but the increasing involvement of general practitioners and also nurse practitioners. This is probably an underestimate of the nurse practitioner role, and that's been increasing encouragingly in recent years. Although the overall treatment numbers have been declining, the proportion of people treated in prison has increased. So back in 2016, only 6% of the total hepatitis C treatment population were in prison at the time of treatment initiation. By 2020, that had reached almost 40%, and the data on 2021 and 2022 are very comparable. So more than a third of patients initiating Hep C treatment in prison at the time. Now that's somewhat damning in terms of the rates of incarceration that we have for particularly for drug-related crime. It's damning in terms of the woeful sort of rates of incarceration we have for our Indigenous populations. But we do take that opportunity within prison to try and provide high quality access to Hep C screening and care. Looking specifically at what impact this treatment is having, this is a survey that the Kirby Institute undertakes every year, led by Lisa Ma, where we sample somewhere between 2,000 and 2,500 people who inject drugs through 50 NSP sites around the country. We ask questions about whether they've been treated with hepatitis C, and we take a finger prick blood test for HIV, Hep C antibody, and in more recent years, also Hep C RNA. You can see back in 2015, only 11% of people reported prior Hep C treatment, and that reached a level above 60% in 2019. With that huge increase in treatment uptake, we see this incredibly encouraging reduction in Hep C RNA prevalence. So active infection falling from more than half of people being actively infected in 2015 to a level of 16% in 2020 and 2021. Hopefully this will continue to trend downwards. With that, we're also seeing a reduction in incidence. So we're reducing the pool of viremic prevalence, so the pool of active infection. You would expect that should translate into reduced risk of new infections. And we compared within this annual needle syringe program survey, many participants have had more than one involvement in the survey. So we linked people that had had more than one participation. And we looked at the incidence pre-DA era versus post-DA era, and that had shown almost a 60% reduction in the incidence from 5.4 per 100 person years. From 13.6 down to 5.4 per 100 person years. So incredibly encouraging. In terms of some outputs from modelling, we look at some of our empiric data, obviously that data on incidence, but also data on the number of new admissions for liver failure and liver cancer that we monitor through our data linkage projects, and we use that empiric data to calibrate our models. And we've been sort of modelling this for many years now. And then we look at different treatment scenarios. So we have a sort of pessimistic treatment scenario where the treatment numbers continue to fall, an intermediate scenario where they're pretty stable, and an optimistic scenario where it increases back a little bit. We can see the pool of people with chronic infection continuing to decline through to 2030, irrespective of those scenarios. you get this commensurate decline in incidence of hepatitis C down to around about 5 per 100,000 per year, which is going to be the new revised WHO target for incidence of hepatitis C. Rather than a 90% reduction in incidence from 2015, the new target will to get the incidence down to less than 5 per 100,000 per year. Treatment coverage should get pretty close to the target of 80%. And the mortality figure, again, WHO is revising their target. So instead of a 65% reduction in mortality, which we wouldn't have achieved because prior to 2015, we're on this increasing incidence of mortality. It's very difficult to turn that around and get 65% reduction. But we will get below the new target, which is 2 per 100,000 population per year. So just a few takeaways in terms of Hep C therapeutic research for people who inject drugs. I think human rights and health access has underpinned our work here at the Kirby, certainly underpinned my work individually. Clinical leadership, investigator-initiated research are intertwined, and we see that in many different areas. And it's really sort of pivotal, I think, to have clinician academics to drive a lot of these therapeutic developments forward and broaden their population access. Innovation's key. It's been really important to try and keep 2 steps ahead of the pack, so to speak. I think we've achieved that in some areas. And collaboration and partnership, though, has been essential. It's been a real sort of pleasure of mine to work with many, many individuals, academics, community members, government over the years of therapeutic development. And it's been, as I said, a privilege to work in the infectious disease sector more broadly. I've been fortunate. I was involved in HIV back in the 90s. My PhD was in HIV epidemiology. I was involved in some of the therapeutic development on HIV front, then very fortunate to be involved in Hep C therapeutic development. I've done a little bit of work in COVID in recent years, not so sort of exciting in terms of therapeutic development, but nonetheless a very sort of meaningful role in terms of broader infectious diseases. So just to acknowledge many of the people in our team here at the Kirby and collaborators, both national and international. Been great working with Jacob over many years. He's been involved in the data linkage work, and he's been a real sort of friend of the Kirby in terms of our work, and there's probably been the odd person in the hepatology community that might not have been as friendly. So it's been great to have Jacob's support over many years. I'd really like to acknowledge Gail Matthews, who's been with us for almost 20 years now. Gail is now my boss at St Vincent's Hospital. She heads up the infectious diseases service there, and she heads up her own program now at the Kirby, the Therapeutic and Vaccine Research Program. Jason Grebley, who's really a leader, not just nationally, internationally, particularly in the area of point of care testing. Been wonderful working with Jason over many, many years. And Pip Marks, who is our clinical trials manager and really was a driver for the operational excellence that we're able to sort of bring to many of these ideas. Has now been poached by Sydney University to be involved in clinical trials sort of development and conduct at your campus. So once again, thanks very much for the opportunity to present today.
Okay, thank you, Greg. So we've got a fair bit of time for questions, which is great. Thank you for a wonderful talk. It's certainly been a very exciting progress, and it's been great being on board for that, but there's still quite a lot of challenges ahead. So I guess so I've got a couple of questions here in the chat already. So given the excellent cure rates that we're seeing with the DAAs, do we know who are the small minority who failed treatment and why? And do we have any sense of reinfection or recurrence rates?
Yeah, look, it's a key sort of question. There's two broad groups, I suppose. There's a group that still do fail in terms of virological failure. That's a very, very small proportion these days. There's people that have more advanced liver disease or higher sort of risk of virological failure, sometimes need sort of salvage therapy. But that, as I said, it's really only 1 or 2% of the people we treat. And in fact, then when you treat them in the second line regimen, generally they're cured. In fact, if we sort of looked at all the people at St Vincent's through the service, we don't have anyone that is still a failure. So some people required 3 regimens, 3 rounds of sort of treatment, but were able to be cured. So there's very, very few patients that that have, a hepatitis C, variant that's unable to be cured with these new therapies. The other and larger group clearly is a group of people that are not adherent to therapy. And we've been looking at sort of treatment discontinuation and tracking treatment discontinuation. And there has been an increase in the proportion of people that discontinue early over time. So there's a significant proportion that get a script for 1 month, don't go back for their second month if it's one regimen, or for their third month if it's another sort of common regimen that we have, a 12-week regimen. So we need to sort of look at strategies to deal with that. I mean, I'm still keen to push the therapeutic development envelope. I'm trying to get a 4-week study going with a combination of Mavorite plus sofosbuvir, because I think for most people that would be a regimen that would provide cure. Hard to get industry to support that sort of study, but I think we may be able to move forward with generic sofosbuvir in addition to glucapravir and preventacir over a 4-week period. There's also an international movement looking at long-acting formulations, so that may be an important sort of downstream sort of therapeutic development. It'd be great if we had a long-acting therapy, particularly for populations where adherence was an issue.
Okay, thank you. Just a reminder to everyone, we've got some questions from the panel here. We still haven't got a lot of questions from the general audience. The Q&A window is open. If we could encourage people to please type some questions for Greg or anyone else on the panel. So what are given, there's been some great results in Australia, but what are the initiatives to upscale these findings to lower resource settings?
Yeah, so it's a good question. In fact, I'm presenting tomorrow night virtually at the Global Hepatitis Summit that's underway in Paris on global viral hepatitis elimination challenges. And there are many. One of the key problems is that we've struggled to leverage investment. If you compare not just hepatitis C, but hepatitis B as well, the level of investment, the major global funder dollars, it's really only a couple of % of HIV sort of investment in terms of care and prevention. We haven't had the community mobilization, the community voices, the community advocacy that's driven a lot of the HIV development and funding. If we look at low and middle income countries and successes with hepatitis C, there have been some exemplars. I mean, Egypt's been an amazing success story where they've screened 50 million people, treated several million people with active infection, and now have evidence of not only reduced viremic prevalence markedly, but really major impacts on Hep C incidence. So they're tracking very nicely in terms of elimination, but Egypt's an absolute outlier. If you look across a lot of the other countries, the other high burden countries, India, China, Russia, very, very limited success in terms of measures, really low rates of population screening. And in those settings, you need broad-based screening. You can't just screen, so-called high-risk populations. Many of the people infected there acquire their infection through unsafe, healthcare-related exposure. So you really do need very, very broad screening, as was undertaken in Egypt. So whether there's the sort of will, the political sort of will and the endeavour to take on something like that is to be seen. And it really does need, I think, a renewed sort of drive to try and mobilise governments to take it seriously as Egypt has taken.
What's the availability of generic drugs in those countries now?
Yeah, look, I think in fact, the treatment side of it's not the big issue. I mean, the cost of generic regimen in many of those countries is less than $50 per regimen. Look, there's a lot of sofosbuvir plus decladisvir still used rather than what would be probably a preferable regimen, but still a highly effective regimen. So I don't think it's the drug costs that's the main issue. I think it's the screening side, because only 20% of people estimated to be infected have been diagnosed. And as I said, the screening sort of initiatives are really modest. The the diagnostic costings need to come down a bit. We need some sort of initiatives on that sort of front. But it does come back to developing national strategies and taking it seriously. And there's just not enough countries that are doing that at the moment. So yeah, I think some of it starts with sort of greater sort of community advocacy and really sort of pushing things from that perspective as well.
Sure. Question, do you think in light of lower income countries, do you think that the new WHO definition of elimination makes it harder for places with low incomes and high prevalence to meet the targets?
Look, I think the revised targets are pragmatic. I mean, they're sort of, you know, the goalpost shifting somewhat. But I think WHO is trying to acknowledge that there are limitations in how we monitor data. I mean, to demonstrate elimination, you have to have pretty good data systems, and a lot of those weren't in place in 2015. So for a country X to monitor that they've had this reduction from 2015 to 2030 without sort of great sort of data is problematic, but they can develop surveillance systems that can monitor the incidence of mortality and the incidence of infection, and they can be developed over time and they can be encouraged. I think the mortality levels that they've chosen are less than 2 per 100,000 for Hep C and less than 4 per 100,000 for Hep B makes sense. I'm broadly sort of supportive. So I think that trying to engage the countries, getting them to understand that this is actually achievable, and working with them to develop data systems and other programs to roll out proven interventions is the way to go. Mark, you're just not coming through there.
And then you kept going mute. I know. I was putting off, it kept jumping back on. I don't know what's going on. Apologies for that. You said you showed the data that we were previously on track to meet the elimination targets for the WHO, but then we weren't. We no longer are. What are the remaining barriers, and what do you think there are gaps in the coverage nationally? What can we do about that?
Yeah, look, I think we lost a bit of momentum, and there's no doubt COVID didn't help over the last sort of few years, but we were losing momentum even pre-COVID. I think that we needed to simplify sort of the screening and linkage to care. And I think that was clear. Look at the sort of the days of people coming into tertiary clinics and being treated, and I mean, they're largely behind us. We see a trickle, as you know, through our tertiary sort of centres. So the answer has to be to not only continue to diversify in terms of the models of care in different settings, and we've done that, I think, better than probably any country around the world, but within the context of that diversification of models of care, to make the sort of testing and linkage to care and treatment to delivery simpler. And that's where the point of care sort of story comes in. It's sort of logical. It makes sense that if you only need a finger prick of blood, you get the result pretty quickly. You can engage with someone, you can use that window of opportunity to try and get them onto therapy, that you're going to do better. So I think we're only just starting, to be honest, with that. So there's a lot of upside. So I'm really quite optimistic about what we can achieve over the next sort of few years. I always tend to be a bit of an optimist, but I'm pretty optimistic that we can. And in fact, we're seeing increased treatment numbers in 2023 in New South Wales compared to 2021 and 2022. It's the first time that we're seeing the numbers of people treated increase. In fact, the last 2 quarters of last year and the first 2 quarters, or the first quarter of this year, have seen an upswing. So we are doing something right. So I think we need to sort of continue those initiatives. And as I said, I'm relatively optimistic. We do need to, there's different subpopulations though. I think someone in the chat, I think Jacob mentioned mental health populations. So there needs to be some initiatives in some of those populations of engagement, and they're sort of a bit ad hoc at the moment. Some services screening mental health key populations. And so, yeah, we just need to keep innovating. As I said, if you stop innovating, you can end up treading water. So if you sit back and think we've got the solutions, then let it sort of roll on. That's when you get into trouble. So I'm really keen to keep trying to innovate even on the therapeutic development sort of side of things, and certainly innovating in terms of the models of care that we deliver.
Okay, great. In terms of the point of care, if you've got, I've seen that you've spread out to high-risk settings, prisons and so forth. Is there a model where you can incorporate point of care into GP practice particularly?
Yeah, it's a good question. You'd have to have, I suppose, a reasonable throughput and a reasonable prevalence. So would it make sense, for example, in a general practice to screen everyone for Hep C when the antibody prevalence is probably 1%? Is that worth doing? Probably not. I mean, the US is going down a pathway of one-off Hep C testing for everyone. And look, that's what they need because they're appallingly bad at everything Hep C. They're not preventing infection. They're not, linking enough people to care. They're not getting to marginalised populations. We're doing a lot of that pretty well. So I've not felt it's necessary to have a one-off screening test for everyone. Our proportion of late diagnosis is continuing to decline. So I think we're doing okay. But you can imagine some primary care practices and community health centres that might have an expected prevalence of 5 to 10%. So in that context, I think embedding some point of care testing would be really useful to look at.
Okay, thank you. Just I guess part of my interest would be, given on this ongoing transmission in the prisons and reinfection, are you aware of any sort of resistant viruses being transmitted? Do you think there's a role? Is that a concern going forward?
My view, Mark, I stated this many years ago that resistance wasn't going to be a problem in Hep C. I got shouted down at a virology conference. I'm still holding to that sort of view. Look, I just think that we're keeping ahead of the virus. And I think the agents that we're using, the regimens that we have pretty high barrier to resistance. And I haven't seen any evidence of transmitted resistance that's going to be an issue. And I really would be very surprised if that's an issue. The big issue for me, though, is that we're sort of not doing anything on the vaccine development front. If I think of what do we still need to sort of do, okay, we can't completely rely on treatment. We're doing pretty well in a country like Australia because we have a good treatment rollout and we have good harm reduction. But if we're going to eliminate Hep C, globally, I think we eventually will need a vaccine. And as you know, there's been very few, only one phase 2 trial of a candidate Hep C vaccine. I'm involved in an international sort of consortium that's looking at potentially advancing vaccine development through controlled human infection model experiments. And it's a really interesting area. There's a community advocacy group of people that have been involved in controlled human infection studies that are really trying to sort of push this agenda for Hep C. And I think it would be one way to accelerate Hep C vaccine development, despite the key sort of ethical issues that you have to sort of work through. I had a discussion with my 18-year-old a year or so ago about the concept. He thought there was no problem with it. He'd be happy to put his hand up. But I acknowledge that there are sort of issues that you have to sort of work through with those sorts of studies. But it's another area of innovation and development that we need to sort of look at to keep things moving forward.
Okay, fantastic. Have we just about out of time? Have we got any last minute questions for Greg? Jacob, anyone else want to ask another question? No, look, I think we've given you a bit of a grilling there, Greg, and fantastic talk. So look, thank you so much. It's been a brilliant talk, and thank you for really great answers to some pretty curly questions there from some people. And thank you. It's been a pleasure having you talk, and really looking forward to seeing some more progress in this field. And great to see you still advocating for the patients and those, particularly some of those communities. Ben, did you want to say anything else?
No, Mark, just from our side as well to thank Greg. It's really, I think, outstanding to see a Australia leading the way in this. I'm very aware of other countries around the world looking at what we are doing here and taking the lead from what we're doing. And thanks for your leadership in this space. Was very informative to hear directly from you.
Thanks very much for the opportunity. Have a great day, everyone.
Okay, thanks, Greg. Thank you.